take the freedom, even the ladies, they can come down and they can start looking at the posters during the break time, and they can have the privilege to interact with these highly cited, distinguished scientists that we have over here. I will call upon our next speaker, Professor Sudhir Kumar. He is from the Center of Evolutionary Medicine and Informatics, a Biodesign Institute from Arizona State University, US. Most welcome, Professor. Thank you very much for being here. Please.
Thank you. So, the title of my talk was a little different from what I put in the abstract book. So I will talk about two things, like what Manolis did. And the journal title of the talk is now Molecular Evolution in Genomic Medicine, since this session has a genomic medicine theme. And ultimately, in my laboratory, in my research, my primary interest is in understanding the patterns and processes at the level of genomes and genes and mutations that has led to the diversity of life. And that includes, of course, species and humans. And we have diverse phenotypes in humans, a number of them that Manolis discussed. And in general, we see around us the tapestry of life is really dazzling and wonderful. And the institute I am in, Biodesign Institute, our focus is also then, when we learn this information, patterns and process, is to actually harness it somehow to use it for genomic medicine, to making human life better. So we have a dual purpose to our research. One is to do basic science, and the second is to see if we can somehow harness this power of evolutionary knowledge, which is primarily coming from comparative genomics of humans and other species, and for the purpose of medicine and health and sustainability. In my case, the focus is actually on medicine in general. So I like to start with giving you a brief overview of what happens, what kind of projects we do in my group personally, and then focus on one or two topics that are of interest to this audience. So there are three major foci in my group. One is the tree of life, but that includes tree from humans all the way to our ancestors, all the way up to the origin of life. And the second is genomic medicine, as I'll discuss today also. And the third is the developmental complexity, which is what is a phenotypic complexity that we're interested in. Now, unlike I think all other speakers that I've heard over the last day or two here, we don't actually generate any new data. We don't have to go out, get tissue samples, or have animals in the laboratory or any of these things. So we do everything bioinformatically. So my laboratory is basically a bunch of computers, high performance, and a bunch of students. And so what we do is analytics. We do analysis of large amounts of data. So synthesis across many studies, many researcher groups, their output, which is really wonderful given the genomic revolution. And the field and tree of life that we are interested in is the phylogenomics, which I'll talk about just a bit today for a few minutes, which has to do with building the history of life, its organization, its timing of emergence, using genome data. In genomic medicine, the field, what I refer to as phylomedicine, is more about how the tree of life, not just the tree of humans, but tree of life in general, can help diagnose disease and adaptation at the level of humans, individuals, cancers, or anything that we are interested in that has to do with humans. And the third thing is what is called evo-devo, evolution of development, where we are very interested in fruit fly embryogenesis, because that's one of the tractable systems, and image analysis, but I will not talk about that today. And when we do this large-scale analytics, we always have to end up developing new methods, estimate how good other methods are, and so we do a lot of modeling, computer simulations, we do analysis of data. So this is a major area for my group. And when we develop all these methods and models and do data analysis to make discoveries, we end up realizing that other people have similar needs. So we develop a lot of software and databases that are widely used by researchers in the world, including software for comparative genomics, software for SNP ranking, prioritization, diagnosis, and software for actually mining image data using images as a primary object. So today what I would do is I will primarily talk about phylogenomics and phylomedicine and give you just two examples of recently published work in what we are doing. And I'm going to start with phylomedicine. This is a term that I use to basically refer to the interface of genomics, evolution, and medicine. And essentially, phylomedicine is not really about what happened in the past, but rather, given what we know about the past, what can we predict about the novel variants we are observing in our genome? And that's the focus in our case. So in a sense, it is different from evolutionary medicine, a field in which a lot of work is done thinking and looking back and saying, why did we get flu? Or when did this thing emerge only? So in phylomedicine, one of the major focus in my group is the analysis of exomes. And as you know, and Manol has referred to, exomes are essentially a complete collection of exons that actually give rise to our proteins. And of course, we have over 20,000 genes which make proteins. And it's a very minuscule part of our genome, but this is the part of the genome that we know a lot about. And even though it is really minuscule, it's still quite big. There are 15 million amino acids in it. So the number of possible variants that we can have in this exome is very large. And in fact, in this exome, the variants that we are interested in have to do with those that change the amino acid sequence. Therefore, they change the protein sequence. And these are called non-synonymous variants, or NS-SNVs, or you can just in short call them SNPs or SNVs. And this is what I'm going to talk about today. We're interested in many other kinds of variants also, but this is the focus today given the shortness of time. And the reason why we are really interested in that is in the last few years, exome sequences has really become very routine and much more accurate than it used to be. So if you look at the trend of discovery of exome SNPs, especially the non-synonymous kind, which make protein change, it really has exploded just exponentially. This when compared to the SNPs that are found in the whole genome, which is also expanding very quickly in absolute number, but it's still a bit more linear than exomes. And it's because exome sequencing is, as I said, routinely done in cancer groups, routinely done for patients for various reasons. And so this is the focus we have, and it's an important focus and an important part of our understanding. So the grand challenge in genomic medicine, and there are many, many of them, and one of them is, if you were to think about individual point mutations being important for disease or make a significant contribution to disease, then how do we identify them? And any individual, you're walking around with as many as 7,000 to 8,000 such variants. Some of them are de novo in the last generation or two. Many of them are family, some are population specific. How are they going to impact, or how are they impacting your health, your ability to metabolize drugs, and all of those things? That's the grand challenge. And so I got interested in this work more than 10 years ago, just doing very simple evolutionary biology, asking the question, what is the human genome variation which is involved in disease, especially at that time, primarily Mendelian diseases, have to do with long-term history of humans and chimpanzees and gorilla, orangutan, all of these species, which is among species. So we decided to just do simple evolutionary biology, try to understand it. And so over the 10-year period from 2000 to 2010, we just tried to develop lots of knowledge about it and made some fundamental discoveries which were, in retrospect, quite obvious. But nonetheless, they were discovered at the time. But during this time, in the last 10 years, there were another group of researchers, unlike us, who were actually developing methods for diagnosing which particular exome variant will have disease potential, or which SNP will make potentially a disease contribution. And really wonderful software like Polyphen and SIFT were produced, which are now used 1,000 times or more than that every year. Today, everybody runs all their variants through them, because you find so many variants and you have to pick a few. And in the last couple of years ago, we began thinking about these kinds of variants and these kind of methods. And we saw there was a new paper that came out in 2011 in American Journal of Human Genetics, which is the Condell software, which took a bunch of these methods together and combined them in a wonderful package, made what they call Condell. It's a hybrid program. And then based on the best features of all these methods, they were able to presumably increase the accuracy. So if you look at the topmost curve, which is a curve which flattens out, goes very quickly up and flattens out, it shows that they could really do an amazing job. They could do true positive, correctly identify disease variants, potentially, 90% of the time, with only 10% false positive. This is an amazing performance. And so it sounded very interesting to us to explore if this is really true. And so we had some understanding of evolutionary patterns. Many of these people actually use sequence alignments. So we expected the things to work out really well for them. So the question I asked very simply was, we asked, if you look at the positions that are most important functionally, that means they have barely changed ever in the history of vertebrates, 500 million years. So evolutionarily, no changes have occurred at these positions at amino acid level. And experimentally, if you look at it, most experimentalists actually look at positions that are unchanged among species because they think this is very important. These are important sites. So we asked the question, what is the accuracy of these methods for these positions? It's a very simple question. And given that the fact there should be 90% accuracy based on what we saw in the literature, we did the analysis. Unfortunately, the results were not really that good. It showed that all of these methods, like polyphen 2, Condell, SIFT, they suffer from severe problems. They incorrectly identify non-disease causing mutations to be disease causing at an astonishing rate of 70 to 90%. So to us, this is quite surprising, and we tried to sort out why this happens, and we did sort it out, and I won't go into detail a lot about that. But what we realized is, okay, so there is such a huge error. This actually means that anybody who used these tools in the last 10 years and picked the most dangerous SNPs probably is wrong more than half the time, or maybe 90% of the time. So this is one of the reasons why we have to really know this kind of problem. So this sort of an evolutionary approach to diagnosing error has not been taken before. And so what we thought is we will develop a method that sort of fixes this problem, because ultimately that is our interest, is developing better methods. And so what we developed is a new method published late last year called EvoD, in which we use fundamental evolutionary properties of the position where the mutations happen or variation is found, and the actual variation in a propensity, biochemical as well as substitutionability. And basically is using 12 primary parameters, predictive parameters. We did what we call, what you might all know about is machine learning and predictive modeling using this data. And we use training data like disease, known disease data, and not non-disease, neutral variants presumably. And we were able to build a mathematical model, a simple one, really regression model ultimately, and it produces a single impact score. It's a number. If a number is 100, it's a very, very bad mutation, like it might cause a disease. If a number is 0, it is really benign, what we call neutral variant. And so if you actually take the 1,000 genomes data that Manol has discussed and put it on x-axis as an impact score and y-axis as the frequency of alleles in the 1,000 genomes data, what you find is a nice result, that there is a negative relationship, and that actually we found that a score of around 50 or so is a good cutoff between truly benign and potentially problematic variants. And so when we use this method, we were able to actually improve upon the error on polyphen and Condell and SIFT, all of those methods. So in fact, the black bars show, compared to Condell, if you compare to Condell, we have reduced its false positive rate from 90% by 70%. That means just false positive is 20% approximately. If you do compare to polyfen, we reduced its false positive rate by 50%. So essentially, around 20% error cannot be removed. But other than that, this method actually takes care of most of the bias that exists in current tools. And this is true for all kinds of variants, high or low frequency variants for the human data that we know about. And I should tell you that we used the same exact data set that was used by Polyfen and Condell for training their models. They're absolutely the same information. This way the results and comparisons are absolutely the perfect comparisons with no changes in data sets. And then actually we applied this to complete exome data for many individuals. We took actually 8 individuals from a Sarah and Egg paper, and we applied this. And if you actually look at positions that are extremely well conserved, which is the left bar, middle one is well conserved, means some change is allowed by evolution over time, means slight flexibility exists, and the last one is a lot of changes are allowed. And if you look at all the alleles found in heterozygous genotype, you find that around 60% of the variants are neutral or non-affected, and around, I would say, 25 or 30% show problematic signs, which is by red coloring. But heterozygote, it seems like, have, across these 3 categories, not much difference when it comes to percent variants. Now, there are more variants found in less conserved sites and ultra-conserved sites, but if you look at fractions, they're pretty similar, and it kind of makes sense because we know lots of variants of functional impact are recessive, and they just sit around waiting to be expressed when they become homozygous. So what happens when they become homozygous is, you see that first of all, homozygous have a number much smaller of bad than heterozygous. And the second is the homozygous in fact show a difference between 3 categories, ultra, well, and less conserved. In less conserved category, there are more homozygous have more disruptive looking variants than ultra conserved. In fact, in one exome, in the homozygous, the deep red you see is just one variant. So at a very high probability of significance, there's only 1 or 2 or a few variants in an exome that are really that bad at ultra conserved sites. So this was actually made sense from the purpose of the way alleles act, dominance of wild type versus recessive alleles versus mutants, I should say, and their recessive nature. So it was a very nice result, reconfirming what we expected. So just to complete on that, if you're interested in using a tool like EvoD, we have recently developed a software, which is what we always do, where you can in fact take your data from any exome survey, thousands of variants, one variant, you can enter that data to this tool, and we have programmed this within a software that we have, which many people use, called Mega, and it's available as a standalone software also, also as a web service. And it also provides you with Polyfen 2 and SIFT predictions. So this way you can see all the predictions, which is not really about just our method, but it's about you having overall good result. In fact, consensus results tend to be very highly accurate across 3 methods. And also we are trying to have researchers in population genetics who are using this variation data also look at this data from the evolutionary perspective, long-term history. So now we integrate long-term history with short-term history in our display or Tree Explorer, where you can examine exactly where that variant occurred, if it ever did, in the history of vertebrates. So this is a software tool that you might find useful for your research. And I should also close this part of the presentation by telling you that we are also looking at nonsense variants. We have a new method for that, which is more accurate than any other method published to date. Insertion deletions, with and without frame shifting, and also even synonymous variants, because we now know a number of synonymous variants are involved. But I will talk about that maybe in the future in any similar venue. So that was the first part of my talk. Hopefully I didn't take too long. The second part is phylogenomics, and this is the part that has to do with the tree of life part. So in genomic medicine, we are constantly estimating when did certain infections occur, when did a HIV strain arise, and all of this requires timing the origin of a strain based on the contemporary sequence. So we have been doing research in this area for a while, and our primary focus in the past has been on species evolution, but the same method can be applied to strain evolution. Ultimately, they are sequences, especially if you're thinking about viruses and bacteria. which have a different sort of a history. So the field of phylogenetics has to do with really trees, using phylogenies and converting them to time trees, where there is a time on the scale. And there are many methods available, multi-diff time, MCMC tree, beast. As you get cancer mutations and all of these data, you can actually have large number of sequences and you want to find out when these mutations happened and what was the time. So you have to use these methods, and people are using them. The problem, as the first I should mention here, is a lot of people are very interested in times of divergence. In fact, if you look at over time, cumulative number of studies published is just increasing every year by a few hundred. So lots of people are publishing. And the number of species for which we know times is almost as many as 50,000. So you can sort of build a tree of life scale to time based on these diverse species. So if you're interested in knowing about any pair of species history, human-mouse divergence time, what do people say? What do the scientists who are working in that field say? And you're a molecular biologist. You can actually go to our website called timetree.org. We even have an iPhone app, an iPad app. You just put in human mouse. It will tell you every study published and what time have they found. And this way you can have a quick look at the field without having to know their nomenclature and their complexities. This will basically give you the information. So it is a lot better than Google, where you have to read the paper. Nowadays no one has time to read the papers, as you know. Everybody's very busy. So this is a resource that you can have a look at. But anyway, the challenge in the computation is in timing of strains or species is it's computationally very demanding. For example, for 400 sequences if you have, the program that many people want to use, BEAST, will take 92,000 days. That's a long time. You need supercomputing square or something for that one. If you had 1,000 taxa or sequences, it will take forever. You just will never finish. So many people just subsample the data or try to deal with it. So the other problem is many tools require knowledge of the rate of how the rate is changing over time in a tree because of the adaptation, because of the strains, because of random nature, and then you have to know when did certain nodes in the tree have a common ancestor to scale everything to time. So what we did is we said, well, what if we don't have any of that stuff? Can we really come up with a method that was fast and easy to use and doesn't require any of this information? And so we did. We published that late last year also. It doesn't require any of the stuff that everybody else requires, which means it is not based on those information. It's more algorithmic than a statistical method, and it's really fast. MC2T, which is the fastest method out there in Bayesian world, ours is a likelihood method. It will take 10 days for 1,000 sequences. Ours will take 3 hours. So it makes it really possible to calculate all of these things very quickly. And this is the published stuff. I won't go into how it is, but essentially our method produces as good estimate as the true times if we have large enough data set. And in fact, for the same data set, you can compare our method to MC2T or BEAST, and we in fact do better because our curves are much more centrally located than MC2T, for example. And if you have a directional change of rate in one lineage, which could happen because of adaptation, for example, our method is still doing well for both inside and outside of that clade, and unlike other methods that are possible to compute, but they have a bias in their methodology. So basically, Bayesian methods, the way they are constructed currently, are slow and actually inaccurate, mostly extremely biased. And actually, we very recently, just the last few months, extended this method where we not only use species times, but also if we know the time of sampling of the sequence. So HIV sequence is sampled from the same patient or multiple patients over time in the last 10 years. That's shown by the x-axis. And then, so therefore, it's almost like you are sequencing ancient DNA. And you want to know when did they diverge from a common ancestor. So in this case, the problem becomes slightly different, but we were able to redo the runtime methodology. And in fact, if you look at the runtime that we produce within a minute compared to BEAST, which takes many days, and the time estimates are practically identical. So it's a very fast solution to produce divergence time estimate for any type of data. And in the case of cancer, that would be very interesting in the future, as you have sequences from different strains, from different parts of the different tumors in the same individual. You want to know, when did this actually get here? When did these cells leave the parent cluster in time? These are the kind of method that people will have to use to estimate that time. And again, we have recently added this to rel time to Mega 6, which is the paper just published in MBE. So you can easily utilize this method in the current version of Mega, which is online as a beta version, will be very soon complete. So this gives you an idea and the error bars on estimating the time of the origin of strains or species. Our method works for both equally well. And so I want to finish by saying that, I use the same exact principles of molecular evolution for all the problems. I have one toolkit I learned, and actually Manolis and I, we both went to graduate school at Penn State at the same time. And what we learned is population genetics and molecular evolution. He does more PopGen and I do more molecular evolution. But ultimately they are two facets of the same problem and the same pattern. And so what we are trying to do here in my group is to work on the field of phylo-medicine, where I insist that long-term history beyond humans is very informative about human variants. It's really an important thing. And evolution should be seen as a technology, not just as a process or anything for solving problems today. So I think of evolution more as a technology than as a field in many cases. And phylogenomics, of course, has solved many of our problems and is going to continue to help us build the history of strains and species and their timing. And ultimately, we are translating this knowledge into practice for practical tools that I think are used widely now by students and researchers everywhere, and even medical fields. So I want to finish by thanking a number of my associates and funding agencies for their support. Thank you.